Good afternoon, everyone. I'm Nick Vigiani, the Assistant Vice President for Research at Rensselaer. Before we get started, I'd like to address a few housekeeping items. First, to better facilitate our program, all attendees will be muted and will not appear on video. This WebEx has a Q&A function, and we invite you to submit your questions at any time during the program. After all 4 presentations have concluded, our moderator will facilitate the Q&A by reading your questions to the presenters. If you wish to contact any of our presenters after the forum, their names and email addresses are listed in the program we provided. Finally, today's webinar will be recorded, and we will post a video link on Rensselaer's research homepage at research.rpi.edu. Now let me introduce Richie Hunter, Vice President for Strategic Communications and External Relations, for her welcoming remarks today. Richie?
Thank you, Nick. It's my privilege to welcome you to our first virtual research showcase. Most people are familiar with Rensselaer as the first technological university in the United States. We have a very rich history of excellence in undergraduate and graduate education and a legacy of groundbreaking research focused on global challenges. We bring together bright, passionate minds from across all disciplines to spark fresh and creative ways to solve the world's biggest problems. This type of collaboration is a cornerstone in addressing global challenges and also one of our pillars as an academic community. Collaboration lights the way for us to innovation, discovery, and creative problem solving. This event is brought to you through a collaboration between the portfolio I lead, Strategic Communications and External Relations, and the Rensselaer Polytechnic Institute Research Enterprise. The collaboration extends to our researchers, faculty, students, and partner universities. Collaboration through funding support is equally important and imperative. This support comes from donors, individual donors, from industry, and from government agencies, state and federal. We could not do the things we do and address the greatest challenges facing this world and educate the next generation of researchers, scientists, game changers without this type of support and collaboration. So today you're going to hear a lot about what our researchers are doing to battle COVID-19 and the pandemic that we are all facing and being challenged by. As a campus community, we have had remarkable results on our own campus in preventing the spread of COVID-19. Our testing, tracing, tracking, surveillance, quarantine, and isolation protocol is robust and proving to be very effective. In fact, as of yesterday, we've conducted 57,195 tests since August. We conduct about 7,700 tests per week. And to date, we have only had 8 positive results, which is clearly remarkable. As I move forward in handing this microphone over to Robert Hull, who is our Vice President for Research for Rensselaer Polytechnic Institute, he's going to present researchers and faculty members who are doing a host of other things to help us battle COVID-19. Robert?
Thank you very much, Richie. Really appreciate the introduction and really grateful to have the audience, sharing this, our excitement for research in general, but specifically how researchers at Rensselaer and our collaborators are addressing this pandemic. So my name again is Robert Hull. I'm our acting Vice President for Research at Rensselaer, and I'm very excited to be coordinating this event or moderating this event. So next slide, please, Jim. So I'm going to provide just a 2 or 3 slides of context. Research enterprise at Rensselaer. We organize our research into 5 signature thrusts, which together enable us to address the global challenges facing humankind. So one of these is biotechnology and life sciences, improving human health and well-being, and understanding better the mechanisms of the human body. A second is in energy, environment, and smart systems, where we are looking to secure sustainable energy futures, both in terms of energy supply, but also in terms of its impact on our planet. A third area is nanotechnology and advanced materials, where we're creating new building blocks for new devices, new systems, and new technologies. The fourth is computation, data, and artificial intelligence, the virtual backbone for understanding complex systems and complex technologies. And finally, media, art, science, and technology, epitomized by our Center for Experimental Media and Performing Arts Center at Rensselaer, or MPAC, where scientists, engineers, and artists come together to create new knowledge and new methodologies. So next slide, please, Jim. So together, these activities, these initiatives enable us to address the fundamental challenges facing humankind in human health, in the natural and built environment, in understanding and mitigating global warming, in securing energy resources and materials resources, and enabling both cyber and physical security. But the clear focus now is on the next slide, the COVID-19 virus. And this is what we'll be focusing on this afternoon. Next slide, please, Jim. This one tiny entity, which has created the largest health crisis for this planet in almost a century. So next slide, please. So this summarizes the broad thrusts of COVID-19 research at Rensselaer. One area is therapeutics, mitigating and finding solutions for the impact of the SARS-CoV-2 virus on the human body. For example, in understanding and preventing the interaction with the spike protein on the virus with receptors in human cells. And Professor Deepak Vashtish will be giving an overview of this area. Also in computation and modeling, applying one of the world's most powerful supercomputers, AMOS, and to predictive modeling and predictive simulation at the national, at the state, at the local, and even at the university level. We are using these simulations and predictions to inform our own testing and tracing strategies. And we'll be hearing several presentations focusing on that area. Supply chain and manufacturing recovery. I'm showing an example here from the work of Professor Jose Holguin-Veras at Rensselaer in understanding how panic buying affects the supply chain. And finally, in the area of personal protective equipment, where we've partnered with Mount Sinai to create a system for UV sterilization masks, which can sterilize thousands of masks a day. We've been developing new manufacturing methods for face shields, and we've created several thousand for our internal community, but we've also distributed 3,000 to 4,000 to local hospitals, and we are in a position to substantially more if and as needed. And we'll also be hearing from Professor Zar and Palermo on a new technology, a new coating technology to extend the lifetime of N95 masks. So if I can go to the next slide, please. These are the presenters. Professor Deepak Vashtish is the director of our Center for Biotechnology and Interdisciplinary Systems here at Rensselaer. He will be giving an overview of therapeutic and other approaches to mitigating the effect of the virus. Professor Helen Zar, Assistant Professor of Chemical and Biological Engineering, and Professor Edmund Palermo, Assistant Professor of Materials Science and Engineering, will talk about a collaboration with Mount Sinai on developing electroactive polymeric coatings which immobilize and deactivate the SARS virus in a mask. Professor Christopher Carruthers at Rensselaer, our director of our Center for Computational Innovations, will give an introduction to our supercomputer. And we are honored to have a key collaborator with us from Cornell University, Professor Harold Weinstein, Director of the Institute for Computational Medicine at the Weill Cornell Medical College, working with our AMOS instrument to advance fundamental understanding of the mechanisms of the COVID virus. And the final presentation will be by Professor Kristen Bennett, Associate Director of the Institute for Data Exploration and Applications at Rensselaer. So let's move straight into the presentations. The first presentation will be given by Professor Deepak Vashtish, again, Director of our Biotechnology Center, and his presentation is Understanding and Tackling COVID from Bench to Bedside. Please go ahead, Deepak.
Hi, good afternoon, everyone. Robert, thanks for the introduction. Next slide, please, Jim. So I have the privilege of directing the Center for Biotechnology and Interdisciplinary Studies, or CBIS, at Rensselaer. CBIS was established in 2004 under the Rensselaer Plan by our president, Dr. Shirley Ann Jackson. The building has a number of different very exciting and state-of-the-art instruments which were funded by New York State. Over the years, we have been able to get funding from a variety of sources, as you can see on the screen, from corporate, federal, foundation, and state. In particular, this relationship between the private and the public partnership allows us to address a number of global issues such as COVID, and I'm going to give you some of the examples of that today. Next slide, Jim, please. So this slide gives you an overview of the kind of research which is going in CBIS. And as you can see, we go all the way from genome to proteome and all the way to population health. Over the years, we have utilized this multi-scale investigative approach to do fundamental new discoveries in the area of protein science and also engineering new protein, which are very useful and relevant for issues such as COVID. We've developed several high throughput platforms. And again, I'll give you some examples of how we've been able to use this to address issues related to the current pandemic. And then going on to bioimaging, a very similar effort in looking at the damages in lung and other areas, developing new diagnostic modalities, as you see on the right, followed by new biologics and antibodies, and eventually looking at various cell-based therapy. So this is a very rich environment which gives us a lot of previous achievement and credibility, which we have borne out to address COVID. Next slide, please. So next, please. So here you see COVID, and we've been, focusing on, as Robert pointed out, that how we can develop therapeutics. So one of the first example is going to be shown here shortly by Jim, which is based upon the fact that our researchers, Professor Robert Lenhart and Professor John Dordick, looked at the heparin activity and demonstrated that heparin binds and blocks infection for COVID. And the initial results, preclinical, have been very encouraging, and we're looking at developing some new approaches where this can be delivered intranasally or through a pulmonary delivery. One of the other examples is shown below here is the work done by Professor Guy Monteleone and Professor Kathy Ryer in CBIS. They started with the structural biology approach and discovered structural similarity between the M protein on SARS-CoV-2 and human hepatitis C virus. And then they looked at variety of drugs which have been used to block the protease activity in the case of hepatitis C vaccine, and 7 different drugs were identified and evaluated computationally, which have been shown to inhibit SARS-CoV-2 and decreasing the replication of human cells. And you can see example of one of those drugs up on the screen. Next slide, please. Following on the heparin story developed by Professor Lenhart and Dordick, we partnered with our clinical colleagues at Mount Sinai and looking at the fact that COVID is also a very important contributor to blood-related disorders, which are seen very commonly in COVID-19 patients. That together we developed a protocol which has been, it's an anticoagulation algorithm which has been proposed by Mount Sinai and adopted widely around the world, to see that depending upon the situation of a patient and their key characteristics in COVID, what are the best therapies? So at what point do we move away from standard anticoagulants to a heparin-based therapy? And you can see the example up on your screen. Next slide, please. So after looking at some of the therapeutics, some work which is being done by CBI in the CBIS looks at how do we understand risk factors and repurposed drug. What you see on your right-hand side on their screen is the work which is being done by my group in collaboration with Optum Labs and Mount Sinai, that we looked at a very large national database of 490,000 plus COVID-19 patients, and looked at what are the risk factors which may predispose an individual to be more selected for an ICU use. And you can see that there are some areas such as the status of glycated hemoglobin, which is a risk factor for an ICU use. On the other side, if you look at metformin, insulin, and glucocorticoid, they would decrease the risk for ICU admission. Next slide, please. So here is an example where we take this approach a little bit further, which combines the knowledge in bioimaging which exists in CBIS with other IWRIs at Rensselaer in a cross-portfolio collaboration with IDEA. And what you see here, that we are able to apply deep learning which on the images which are obtained from the CT chest volume, and then apply it in conjunction with data related to demographic, blood tests, and some of the vital signs into a predictive model by selecting features and predicting outcomes. And at the time of admission, this algorithm can predict whether a ICU is needed or not for the patient, causing a very important clinical decision to be made earlier, saving a number of lives. And this protocol has been executed with our collaborators at MGH and other Boston area hospital. Next slide, please. So here I'm giving you some examples of what else we are doing in the area of tackling COVID-19, starting with plasma therapy. This is a outcome of a public-private partnership under NIMBLE, which is the National Institute for Innovation in Manufacturing of Biopharmaceuticals. Together with Johns Hopkins, we are developing a high throughput platform to identify the sera needed for the plasma therapy. This will help enhance the plasma therapy and make it much more effective. This is the work done by Professor Steve Kramer and his group with Johns Hopkins. Next slide, please. So we all are beginning to think optimistically forward that we will have very specifically new therapeutics, monoclonal antibodies, and vaccine to tackle SARS-CoV-2. The question begins is what may be the best time of the day to deliver these drugs and treatment in order to optimize it. So the work done by Professor Jen Hurley in CBIS has found that both inflammation and the ACE2 levels, which have been involved on the SARS-CoV, are actually subjected to circadian rhythm changes, as you can see on your screen, at the different time of the day. So based upon this information, her group, working with Professor Kofus, is developing an approach as to how and when to give these vaccines and other treatments in order to optimize their effect in tackling COVID-19. Next slide, please. And this is the example which my colleague Robert Hull alluded to, is the work we've done with collaboration with Mount Sinai, where we're using UVC to kill SARS-CoV-2. This has been applied in the context of personal protective equipment. And you can see here on your screen between the two that 120 seconds of exposure has been able to neutralize SARS-CoV-2. And on the right-hand side, you see that this dose has been very effective. So we are now working with Sinai to seek further approval for this process so that we can bring this technology to the bedside. And I would like to end here and pass it back to my colleague, Professor Helen Zha, and to Professor Robert Hull.
Yeah, so I'll go ahead and just briefly introduce the next presentations given by Professor Helen Zha and Professor Edmund Palermo, self-disinfecting face masks to prevent the spread of COVID-19. Please go ahead, Helen.
Thank you, Robert. Thanks everyone for joining. I apologize ahead of time if my voice cuts out. I think my internet may hopefully cooperate with us. I'd like to begin the conversation by setting up the big problem that we face in COVID-19 when it comes to protecting a person, particularly a person such as a healthcare worker who often comes in contact with many patients of COVID-19. The big challenge in a disease like this is, in addition to being a highly infectious disease, COVID-19 can be transmitted by airborne methods, by airborne droplets specifically. And so whenever any one of us talks, sneezes, coughs, sings, or yells, we always emit from our mouths a variety of aerosol droplets. In the case of COVID-19 patients, those would contain live virus. And the droplets that are large, so more than 5 microns in size, generally settle out of the air onto surfaces, in which case you would want your sanitizing wipes and all of that. But it's the droplets that are smaller than 5 microns. And to give you an idea of what 5 microns is, a human hair is 60 microns. So 5 microns is about the 10th the size of a hair. Those are invisible to the naked eye, and they hang around in the air for extended periods of time. And it can sometimes even be the case that the viruses in those droplets, the water around them starts to evaporate, and at some point you just have one or maybe a few viruses hanging around in a droplet that's much smaller than 5 microns in size. So the COVID-19 SARS-CoV virus is approximately 100 nanometers in size, and as a standard of comparison, that's 1,000 times smaller than a typical human hair. And so one of the challenges in personal protection, in providing protection for healthcare workers, specifically against being infected by COVID-19, is how do we protect them from exposure from droplets or viruses that are in air that is so small. Next slide, please. And one of the key pieces of personal protective equipment that is needed are these masks. They're called N95 face masks or N95 respirator masks. These masks are rated to block out 95% of all particles in the air from reaching, in this case, the person's mouth. And for healthcare workers, this is a very critical piece of personal protective equipment. And how these masks work is that they contain within them, if you actually cut one open, you'll see there's a couple layers of what looks like fabric. And usually the middle layer or the middle 2 layers will have the filter layer. There'll be the active filter material. And what these filters look like is, I have a large electron microscope image here in they look like a bunch of spaghetti fibers all tangled upon each other. But these filters are made of polypropylene, which is a plastic, but very, very small polypropylene fibers that are it's a non-woven material, so it's melt blown, so it's not like your typical fabric. And these fibers are so small and so densely organized upon each other that they're able to provide a filtration ability against even something as small as a virus particle. So that is one method that these masks work. Now, on top of this physical barrier to viruses going through the mask, there's an additional challenge, which is that when you have a filter that's so dense, you start running into the problem where you're actually decreasing the air permeability through the mask. And that's why, actually, if any of you have ever worn an N95 mask, it's not the easiest thing to breathe through. But considering it's blocking out 95% of all particles, it's actually engineering accomplishment that we can breathe through them. And one of the technologies that enables air to pass through relatively easy through one of these filters is that on the surface of these very small filters, there is an electrostatic charge called an electric charge. And these actually help improve the ability of the filters to capture particles, including particles, but maintain breathability through the mask. Now, where this brings a challenge to our current COVID-19 pandemic is, if we go to the next slide, please, Jen, is that these masks, because of the way this fiber technology has been developed, they have no inherent ability to kill viruses. They simply act as a trap, but you still have live virus trapped within this mask. And because of the particular technologies, this electrostatic charge and the small polypropylene fibers, they really can't be disinfected with your typical solution-based disinfecting technologies such as waving or dunking in alcohol or dunking in bleach. And the way they're developed this technology is they're intended for single use only, which means that the doctor or the nurse should discard their mask after they see every patient to avoid taking that contaminated mask to be in contact with the next patient or in contact with somebody else. And what that set us up for as a society is a shortage of these N95 masks, which is a problem. It was a very, very large problem back in April. It's still quite a significant problem now. And so the solution that we propose in this project that we're working on is to develop a way to coat the filters of these masks with a polymer that is actively able to kill viruses on contact. And in this way, this will allow these masks to be worn for more extended periods of time without having to discard them in between each patient, as well as allow them to be potentially disinfected and reused without worrying about decreasing filtration efficiency. And at this point, I'm going to turn it over to Ed, my co-investigator on this project, to describe our project in further detail.
Okay, thanks, Helen, and good afternoon, everyone. Thanks for joining us. So on the next slide, I'll talk briefly about how we're thinking about approaching this problem. And in fact, we are currently making some great progress. So we want to take a material that we know is antimicrobial. So it can kill a broad spectrum of bacteria, and it can deactivate enveloped viruses such as SARS-CoV-2. Now, such materials are already known. I've worked in the field of antimicrobial polymers for about a decade now. And so we have lots of formulations that we know can kill microorganisms. The problem, though, as Helen pointed out, these masks are porous materials made of very tiny fibers. And so we want to coat the surface of every one of those tiny little fibers with a really, really thin but dense layer of antimicrobial polymer. And so you can imagine this is an engineering challenge, because if I put too much polymer down, I can clog the pores and then you won't be able to breathe easily through the mask. So it's already difficult to breathe through an N95. And so we want the minimum amount of really ultra thin coating on the surface of these fibers to get really potent antiviral activity. If we're able to accomplish that, which experiments are currently underway, what that would mean is that when the virus, when aerosols contact the mask material, not only is the virus trapped or filtered, it also gets directly killed. So this mask would be inherently self-sterilizing. So the way we're approaching this is by a solution-based coating process. We submerge the material in a solution containing the antimicrobial polymer. You have to really precisely control the conditions. So the type of solvent, the temperature, the time, the concentration. And if you get it just right, you can form this kind of thin conformal layer that we're aiming towards. And so we've had some success in recent weeks, and hopefully we'll have a prototype fairly soon, I think. So then on the next slide, we just want to mention how this works. So basically, the polymers that I work with they have a positive charge. And so that enables an attraction with the negatively charged outer envelope of the virus. And these polymers also contain some hydrophobic groups, so they don't like to be in water. The lipid envelope is also hydrophobic. And so much like a soap molecule can disrupt membranes, these hydrophobic and cationic polymer surfaces are also putatively able to strip away that outer envelope on the virus, and that eliminates the ability of the virus to infect the human cell. So it's essentially, you can think of it as being dead. Another approach with a different kind of polymer stuck on the surface would be this multivalent binding interaction, where we grab a hold of those spike proteins and we don't let the material go. And so once this happens, once again, the virus would be unable to infect a human cell. So we look forward to answering any questions that the audience might have. And we, of course, want to thank our sponsors and some companies that helped us out. Well, the materials were supplied by this company, Hills Inc. Our collaborators at Mount Sinai will be testing our prototype against actual clinical isolates of the SARS-CoV-2 virus once we finish our work here on campus. And thank you to the NSF for funding.
So thank you, Professors Zar and Palermo, for a fascinating story, and we're really looking forward to the results of this work. If we can now introduce our computational focus. This is a joint presentation given by Chris Carruthers, Director of our Supercomputer Center here, Center for Computational Innovations at RPI, and Professor Harold Weinstein, Weill Cornell Medical College, who has one of the very most exciting projects on this computer. We're honored that he's joining us today. So I'll pass over to you first, Chris.
Thank you so much. Thank you, Robert, for the introduction. So the first thing I want to do is start off with an introduction, if you will, to our computational resources we have. In particular, the AMOS supercomputer is the 26th fastest supercomputer in the world and the 5th most power efficient in the world. It's actually capable of performing over 8 quadrillion operations per second. That's an 8 with 15 zeros behind it in terms of math operations per second. To put that in perspective, that's the same amount of compute power as if every human in the world, all 8 billion people, were performing 1 million math operations per second, all at the same time. Now, this system was actually designed by IBM, and it serves as a test bed for the New York AI Hardware Center. This is a strategic partnership between IBM, New York State, Rensselaer, and several key industrial members. The center's goal is to design and test next-generation hardware that will accelerate AI and machine learning tasks. However, in response to the COVID-19 pandemic, IBM, Rensselaer became founding members in the National COVID-19 HPC Consortium, which redirected vast U.S. supercomputing resources towards the goal of accelerating our understanding of the COVID-19 virus and the development of treatments and vaccines that will help address infections. To date, over 150,000 compute node hours on AMOS have been allocated to COVID-19 projects from teams at the Cornell Clinic, Harvard, IBM, Northwestern, Rensselaer, and Cornell. Today, we highlight some of the groundbreaking computational work on AMOS done by Professor Weinstein's group at the Cornell Medical College. Harald, I'll take it over from here. Thank you.
Thank you very much, Chris. To begin by telling you about that has become possible only with the use of AMOS that Chris so ably described. I bring you back to Professor Hull's description, if you want, of what we know about the coronavirus or how we have visualized it in order to illustrate both what we knew and what we didn't know in pursuing our project. We knew that the ball that you see here in red contains the genetic material of the virus and has to enter the cell in order to release it. That is the mode of replication. We also knew that the skin of the ball must fuse with the cell membrane, but we didn't know how. We knew that the spike attacks the cell to allow the ball to enter, and we knew that it's a specific part of the spike that gets to pierce the membrane, but we didn't know how. And that is what our project went to find out and to use in fighting the entrance of the virus into the cell. So very briefly, you see here an enhanced view of that ball and spike that tells you how these spikes are used to connect with the human cell. What you see here is that the virus has evolved to identify something that exists in the human cell, a cell receptor called ACE2, and in its structure of the spike has created these receptor binding domains shown here in cyan, which attach to this cell receptor and brings the virus very close to the membrane, but not close enough to pierce it, not close enough to connect with it. And this was one of the greatest surprises in the scientific community, is how the virus manages to send fusion agents that allow it to insert into the cell membrane. What you see here is that the virus has done something extraordinarily clever in evolving to infect us, and that is to hijack an enzyme that is right close to that cell receptor. And what this enzyme does is to cleave the spike in one particular place, just one snip at this particular place called S2. And what this does is described here from detailed structure and massive computational work that we did on AMOS to find out what is happening between the time that the enzyme cleavage has not happened and the entrance of the virus. So you see here that this fusion agent is closely packed to the structure of the virus. But what you see here is that immediately after enzyme cleavage, this fusion agent, the fusion peptide, begins to separate from the structure. And that separation triggers an extraordinary rearrangement of the virus, where this entire part moves down, as it were, from this place and positions these fusion agents as darts to being ready to penetrate the membrane. Because we found this out, we were able to use the computer in the really massive simulations to find out exactly the main mode of insertion of this fusion agent into the membrane, what it looks like there, and what it does to the membrane. These are extraordinarily important elements in our ability to move to the next step, which is where the project is right now, with some very interesting advances in results. And that is again feasible only in the collaboration of our design work with the computation on AMOS, because we are using artificial intelligence and the computational modeling, which we call simulations, because it goes through a what-if scenario, as I illustrated to you in the previous slides that Professor Hall mentioned, to discover compounds that stop the fusion peptides of the spike right here from entering. That is possible only because we know what the spike looks like when these fusion peptides have penetrated the membrane. And this information is shared with the group of Dr. Payal Das at IBM, and the novel artificial intelligence framework that she has developed, which combined with our work, allow us to identify a whole series of completely new kinds of compounds that will stop and that can and do stop the spike from entering the cell. And this is where we are now for the benefit of everybody. And I hope you wish us luck. Thank you very much.
Well, thank you, Harold and Chris, for a truly inspiring presentation. I mean, we're getting down to the real molecular mechanisms and the molecular cure, well, the molecular approaches. So thank you both. So our last presentation, before we move into a question and answer session, will be given by Professor Kristen Bennett. And the title is Tackling COVID-19 Using Data Analytics. Please take it away, Kristen.
Okay, thank you very much. So I'm reporting results on a program by United Health Foundation to create our next generation of health informatic researchers. If you can go back to the beginning, that would be good. And so our goal here is to examine the pandemic and see how we can address it using data analytics. Next slide. So we have teams of undergraduate researchers with a little help from graduate students. There's been over 100 of them to date since March, and they're creating applications that enable us to understand the COVID pandemic. So specifically, our first one was called COVID Minder. It monitors daily COVID cases and mortality and looks for disparities. So on the right, you can see that different case rates in the United States in different states, and some states have much higher rates than others. So we want to understand, why does it matter where you live for your COVID experience? So next slide. So what we did was create an analysis of the socioeconomic determinants of COVID. We want to understand, why do some counties in the US have higher rates of COVID-19 deaths than others? And this is controlling for the fact that it hits different communities at different times. So unfortunately, what the analysis shows is that there's great health inequities in the United States. So communities with minority population, immigrant populations have higher risk of death from COVID. And we also found effects from other things, the quality of the air, the access to healthcare, and many other factors. So this can help inform interventions to help us have better outcomes in the future. And they actually published a paper, our undergraduates did. Let's go on to the next slide. So the next team decided to look at the pandemic in real time using Twitter. So they wanted to try to use AI methods to understand the conversations and sentiment about mask wearing in Twitter. And they collected over 1 billion tweets. They're still collecting them all the time related to COVID. They extracted out what was going on. They found groups of sentiments and groups of clusters that on different topics. They characterized those topics and looked at how they were evolving over time. And this is important because we can understand the impact of public health information, and this can help us see how are they doing and what can we do to improve their impact. So we can use this in the future. And this results in a paper. You can click on the link and see it. Next slide. So another team of students tackled the issue of how do we safely reopen campuses? And they created an app that's publicly available called COVID Back to School. And what it does is it uses mathematical model informed from all this data we collected in COVID Minder about the rates and cases. And you can predict for a given school, how might infections evolve under different scenarios. So you can look at, how much testing should I do? What kind of social distancing should I have at a school? And this was actually used to inform RPI's decision making on their testing strategies and opening strategies, which, as we heard before, have been very successful. And it's available for high schools or boarding schools or universities or any institution to use. Next slide. Another thing they decided to tackle was making campus safe. So in real time. So we created an app called RPI Campus, Safe Campus, and it's really directed to the administration. And it uses Wi-Fi access point data, which monitors, how many people are in various locations. Everybody's got devices. And we use this to see if there's crowded spaces or too many people in too many places. It's a great application because it's completely private, because it's only based on counts of users. We don't need any individual information about the users. So this enables the administration to see what's going on in real time on campus. And a neat thing about this app is that it inspired another app. So the students working on this project last summer said, hey, we could use this to help make the student experience safer by helping students make good individual choices. So next slide. They created an app called RPI Study Safe. And it answers the question is, where should I go to study? So that, we have students on campus now, but, where you should study outside your room isn't always entirely clear. So they made this app that monitors individual buildings and enables you to see how close they are to capacity. So you can pick which buildings to go to at given times. And also uses predictive algorithms to decide, how busy are buildings likely to be in the future, so you can plan your day. So we're actually releasing this app in the next couple of weeks, and it'll be able to help students have a safer experience on campus. And it was totally created by the students. So in conclusion, our students in the Data Insight Lab have been creating impactful solutions to the app, to the COVID pandemic. They've been helping us understand what's going on. And at the same time, we're creating the next generation health informatics researchers. And they're learning to work together in teams remotely on important issues. And this is exactly the skills that we need for students going forward. So thank you. I'll be happy to answer any questions.
So thank you very much, Professor Bennett. And I think this epitomizes the inseparable link between research and education at a university such as Rensselaer. We work closely with undergraduates and research experiences. We view as an integral part of their development as scientists, engineers, and business, and humanities and sciences. And this is an example of our undergraduate population really getting engaged through Kristen's leadership in questions which matter to all of us. So thank you for that outstanding example. So I'd just like to wrap up the presentations by saying we hope this gives an introduction to some of the activities at Rensselaer and our collaborators in New York State. It's really just the tip of an iceberg. We have literally dozens of projects ongoing at Rensselaer to address the COVID virus and its impacts. So please continue to follow our progress on our website, and please reach out to us if we can answer any broader questions or any suggestions you have beyond the discussion session we're now just about to have. So what we'll do now then is encourage all of our attendees to start posting questions on the question and answer site, and we'll try and deal with them as best as we can in the real time. But as those questions start to come in, I would like just to lead off with a question or two. So first of all, I'd like to ask Professors Carruthers and Weinstein, what we are doing with supercomputers now is truly extraordinary. I mean, how does this compare to what we could even have done 4 years ago? I mean, that's the lifetime of most of our undergraduates pursuing their degrees. I mean, if you take the time a student enters their undergraduate degree program in most disciplines to the time they finish in 4 years, how much have things advanced in those 4 to 5 years? Chris and Harold, if you can answer that question.
Yes, I will be glad to address this because we are right there at the bleeding edge of this technology that it has always been the part that stopped our dreams short of turning into quantitative reality. Biology is growing in front of our eyes because of the structural information, because we are now able to visualize these enormous structures that nature has put together in atomic detail. But when we look at them, they're frozen. And what they do is to move around, adapt to their environment, and perform functions that we cannot really just think out. We have to understand them at the level at which they happen in the environment that happens. That is only possible with massive computational power. The other side of this coin is that the moment we have this computational power and the moment we gather this information, we are now avail ourselves of an enormous amount of data points, which computer science now allows us to analyze and find the smallest differences or the smallest types of similarities among, for example, viruses that we talked about. So now we are in our hands, we have a description of the relation between the COVID-19 virus, the SARS-CoV-2, and the viruses that plagued us before. But more importantly, my group and a large number of scientists are involved in doing what-if modeling on the computers based on this data to tell us what other viruses of this kind could come and create pandemics in the future. And we can prepare now to stop this. This is all computational power based because the analysis that this allows us to perform is very far reaching. And it is correct that Amos is one of the major supercomputers. There are very few of these that are available directly to science and to scientists. There are many of them that exist, but they are put to other kinds of uses. And therefore, the contribution of what Rensselaer is doing in this field is irreplaceable and really unique.
So thank you, Harold. Let me turn to you, Chris, because you always have this some lovely answer, Harold. Thank you, but Chris, you always have like this great analogy, which sort of helps us understand just how much the doors have opened. So, do you have something which to compare now to 4 or 5 years ago in a nutshell?
So when our center opened, our first supercomputer was an IBM Blue Gene L, and you can imagine a Blue Gene L, one rack. The compute power of one rack, and it was a very large rack. It was actually slanted because of the air-cooled design, and it was a very large rack. But think of what you think of as a rack of computers. So going back to 2007, all of that compute power for 2007 has now been compressed down into a single card of compute called a graphics processor. So we're talking probably 1,000x compression. And so the students that were taking my parallel computing course then and go forward now, they actually having 1,000x more compute power. And as Harold's pointing out, this is taking his computation and everybody else's calculation to this next level, to where we can be not just figuring things out sort of in the behind, but now we can be a lot more predictive in our analytics about what we want to do and model for the future and not just what's happening today.
So thank you. Those are wonderful answers. So we have a question from our Dean of Engineering at Rensselaer, Professor Shekhar Gade. And his question is, can you comment on how much the virus has mutated since inception of the pandemic? And how does the rate of mutation then impact vaccine development? And I think I'll pass that to you, Professor Vashtish, for the first pass at the answer.
Thank you, Robert, and thank you, Shekhar, for that question. The rate of mutation for this virus is fortunately low, so that does affect positively in terms of the vaccine development. In terms of what is the rate of mutation and how much it is sort of done across the global part, maybe it's the data-centric question, which either Chris or others or Harold can answer. I'll pass that on there.
I'll be glad to give it a try, and I can tell you that the scientific community is following this with enormous interest. There is a very interesting element in the development of the vaccine that has not been exploited before because of what we now know about the structure of the virus, which, again, I'm emphasizing, we didn't have that kind of information for other viruses. And this is what I want to tell you. The virus, the protein of the virus, is literally covered with a sheet of molecules of sugar. I'm going to call them that. So for the layperson, I'm not going to say glycosides. And these have evolved on the viruses in order to protect them from antibodies. And they were very effective. But because we know the structure, the new antibodies incorporate these kinds of elements of the sugar envelope in what the vaccine recognizes. And their mutations don't help because those structures stay the same. They are a particular kind of envelope. So I guess we managed for the moment to trick the defenses of the virus with antibodies that go there. That is number one antibody kind of effect. But the vaccines actually are of several kinds, and two of them at least are directed to what happens to the virus already in the cell the moment what I told you, the genetic material begins to be replicated. And it has to do with attacking the material of replication, the RNA. And so these are much more sophisticated vaccines. That is why they were at the beginning seen with some circumspection. But the two of the prominent vaccines are of that kind. And those are not subject to the protection of the virus by mutations. The mutations that we know of, the famous D614G mutation that is now prevalent in all the viruses that infect us in the USA, has allowed the virus to infect faster. That is a form of protection because it fights the antibodies that would come outside the cell. But it is not protection from what happens inside the cell. So as you can see, we are fighting, but we are now fighting with knowledge. Sorry.
Yeah, that's a beautiful way of putting it. Thank you. That is a great punchline, Harold. Really appreciate that. So there's a very interesting question, which I will perhaps ask Professors Zar and Palermo to take the first attempt at. It's a very fascinating question. Are there silver linings you all see as being researchers at this time to be able to help humanity in such difficult times? So are there things which are positive outcomes of our response to this virus? So Professor Zar, perhaps I could ask you to answer that question first.
Sure, thank you. I think it's a scary time for all of us, but it is very, I think it's positive that we have the expertise and the knowledge and the resources to go after all of these, projects to combat COVID-19 from various areas, everything from the molecular level to the materials science technology aspect. And I think one positive silver lining is that in doing so, the students at RPI, undergraduates, graduate students, are exposed and involved in these efforts. And so if there's any, I think one of the presentations we saw today was a great example of this is a great training / educational time for the students that are involved earlier. I think it's probably one of the more rewarding aspects of the research, for me at least.
Thank you very much, Professor Zar. Professor Palermo.
Yeah, I would definitely agree with that from the educational standpoint. We always tell our students that learning science and engineering is so important for real world applications, and now they're seeing it happen in real time when the stakes are very high. So I do feel that way. And I would also point out that, many of the technologies and the developments that occur during this pandemic time will continue to be enormously useful even after COVID is behind us. And so if you're thinking about PPE and making it last longer, as one example, and some of the computational things and the data analytics that we heard about as well, those will have far-reaching applications well beyond COVID. Yeah,
that's great. And I can't resist adding a comment myself, because a similar question came up a few months ago at a panel I was hosting for our alumni in the northeast of the country. And there were two responses which really struck me. And one was, this is an opportunity for a shining moment for science. That it can be the case, and hopefully will be the case, that not only will science be the solution to this pandemic, I mean, that will presumably be what will unfold, but that the public will understand this, and that this is something which can transform, further cement the appreciation for scientific research in the general community. And the other comment I heard, which was even more specific, but was even more, in some ways, inspiring, from somebody who worked as very senior in the medical device field. She said that, to me, this is a turning point. I feel that the general public finally understand what medical technology is about. And we're seeing that all the time, right? I mean, although cases in the COVID virus continue to increase, the number of deaths is thankfully not keeping pace. I mean, at least for now. We are learning so much about how to treat, and the technology and the therapeutic approaches even now for mitigating the virus, that, you know, it's just, it can be an inspiring time, and it can have lasting effects, I think, not just in terms of what very important things Helen and Ed talked about, but also in terms of, I would say, unifying different aspects of society. And that would be a wonderful outcome. So there's another great question here about when do we think this wonderful research and solutions, and I've got to just scroll down to get the rest of the questions, sorry, I had read it earlier, will be implemented. So what is the time frame when this is going to make a difference? And I might ask each of the presenters to give a very short answer. I mean, just a few sentences. What is the timeline for this making a difference? So I'll start with you, Professor Bennett, if I may.
We're leading the charge, right? Because as we get, we can do stuff now. Yeah. So, you know, it's been really exciting to work with the students, and let's make an app that we can put out this semester. Let's make tools that help us decide right now how to open campus. So you're going to have a very immediate effect. So
I can't resist an anecdotal comment then, that at a few times in the past 6 months, I've contacted Professor Bennett and said, do you think X is possible? And next thing I know, I open up the website and the answer to X is there. I mean, this has been remarkably...
I can give another example of that. So the COVID-19 and Diabetes Summit called me up and said, can you give a talk on the social determinants of COVID? And I said, well, you know, I did the social determinants of mortality. I don't see why I couldn't do that. So I published that paper, you know, in 12 weeks. I gave the talk in August. I didn't even know, you know, like, so, but you can do it. It's great.
So then I'll move on next to Professor Vashtish. Of the different therapeutic and biomanufacturing approaches you were talking about, Deepak, what is the timeline there, do you think?
So Robert, some of the things have already happened. I, you know, shared the anticoagulation protocol, which has made a very immediate difference to a large number of COVID-19 patients. So that's it's a good example of what we can do right away. We're making progress on the plasma therapy, looking at the high throughput part, talking about probably 3 to 6 months in terms of, you know, delivering on that project. In terms of the UVC, we've already shown the result that it's sort of working. We are approaching the FDA to get an emergency use authorization through Mount Sinai on that one. So again, that's something which, you know, can be, you know, anytime depending upon, you know, how FDA reviews it. So, you know, a lot of those approaches are very direct, and the same example for the hepatitis C blockers, you know, the inhibitors for hepatitis C, you know, protease blockers, that they exist in the market, and, you know, there's a repurposing part. So I'll stop here, but most of them are within 3 to 6 months.
Yeah, and this is great. So far we've seen timeframes of now to 6 months, which is, you know, very exciting. Now, Professors Palermo and Zahar, what would be the response for the work you're doing?
Thanks. So I think it's always hard to predict the pace of research. We are aiming to have prototypes within the next few months. But I do want to stress, we want to do our due diligence in both making sure that the masks are able to deactivate viruses and also to test against NIOSH standards to make sure they retain their filtration efficiency. But one of the reasons that we went with a coding approach rather than going back all the way to the point where the filters are made is that we wanted to come up with a technology that can be applied to pre-existing already manufactured masks. So you don't have to go all the way back into the manufacturing line all the way in the beginning and interrupt there. You can, once we get this technology nailed down, essentially take it and apply it to stocks that people already have on hand. And we're purposely looking at polymers that are not overly expensive, that are not overly rare, some are commercially available, and looking at very gentle solvents that can be used without, big equipment or fume hoods, with the idea that this can be ideally implemented by almost the end user, and so decrease the amount of time it takes to get it into the hands of people who need it. So all of this was in consideration when we designed and conceived of the technological approach.
Thank you so much. I mean, Professor Palermo, would you add anything to that?
No, I think that Helen covered it. We'll get it done as quickly as it can be done, making sure that it's done right and it's going to be safe and effective.
Yeah, that's of course so important. Thank you. And then Chris and Harold, I mean, what would you estimate as the timeline for impact of the work you are both doing?
May I say that I have an answer that may be surprising. The time is now. It's ongoing. We are all familiar with the warp speed project for the vaccine. It is warp speed, but it is not generated by rhetoric. It is generated by the fact that I now have 3 former students who have generated data at 3 different companies that allowed them to engage in this warp speed, because there is no precedent to the creation of 3 different types of vaccines at the same time in 3 different parts of the world, and to say nothing about other institutions. And that is all based on what was generated 3 months before. So the answer is, it continues. Everything we do at any point is immediately incorporated. That's what is beautiful at this time in the engagement. When it will go out to every person in the world is another story, but without that, what is actually happening at this point would have been just unthinkable. And if you remember, Dr. Fauci thought it was unthinkable.
Yeah, well, thank you. I mean, that's a great perspective. And Chris, would you have anything to, you've got the final word on this question, on this really critical question. Anything you would like to add?
So I'll key off what Harold was saying. I think now is the time, and what's interesting, at least I can say from the computational perspective, as well as storage technologies, and the fact we're on this WebEx call, all of these things have sort of come together to be an enabling technology to really sort of solve the COVID-19 pandemic in a way that I don't think we really thought we could do, and we sort of have, and we sort of marshaled resources together here at Rensselaer and nationally to really address this. Again, you hate to hear the phrase, but it truly is the unprecedented situation we find ourselves in.
So thank you. And I've actually going to just got one final question. There's been a lot of good discussion, between the audience and the individual presenters. Thank you all for that. The final question I want to ask is to Professor Bennett. Coming back to this question of the integration of research and education. So with all that you have achieved, Professor Bennett, how important has the ability to work with talented undergraduates been in realizing that work?
Oh, totally important, because I had this program, I had all this human resources of incredibly talented students to try all these things very rapidly. And I didn't have a particular agenda in mind when I got that grant from United Health Foundation. It was a very generous grant. They just wanted me to make students engaged in health informatics careers. And it's brilliantly successful at that. I mean, I'm writing so many graduate school letters because the students are so engaged with COVID, right? I mean, they're just like, oh my gosh. And I wrote this paper and we've got them, and also it worked out well that we're teaching them. We took our technology for remote collaboration and research, and which we're quite good at because I collaborate with people all over the world. And I moved that right over to education, right? So our research undergraduate teams didn't skip a beat. We've got them engaged at, working on teams with using this new technology. So I think the research really benefited the students, and the students really helped the research because we could do all this thing. And it's really amazing that we've gotten to the level that they do really impactful stuff. I mean, they're writing papers, they're making apps in weeks. And I think the great thing about working with undergraduate, they think they can do anything, and they really can. They're not limited in their minds. They just want to go. It's wonderful.
So like most major research universities, our engine of innovation is our graduate student body. But integrating the undergraduates into our research mission is something we have in our DNA here at Rensselaer. And it's just so exciting. I think you said 112 undergraduate students. Was that the number in your first slide?
Yeah. So we had different programs that I didn't talk about, but we had biology programs on the source of the pandemic from the biology. It was, due to deforestation and things like that. I didn't participate in that. And we also had programs for incoming RPI students, minority students who got to do a little bit of research. So I'm including them in the numbers. And our core that we're doing the research I presented here is probably about 60, 70 students.
All right. So I think we want to celebrate what's been a terrific event with the excitement and ingenuity and sheer brilliance I think our presenters have demonstrated. We've had great questions and participations from the audience. Certainly want to thank very much those people such as Chris Nolan and Nick Vigiani and RPI here who've spearheaded this event. I want to thank all our presenters. And Jim Evans from Multimedia Services has been keeping this all running behind the scenes. I apologize for anybody else I've forgotten, but most of all, I want to thank the audience on this presentation. I mean, we're doing this for you, and it's so delightful that you're here, that you're interested in our work. We are, most enthusiastic to share our work with you at any time in the future. And I'll pass over to Richie Hunter in a second, just for any further sort of concluding message she wants to give. But the last thing I want to say, of course, is that I wish you and all your families and colleagues and friends the best of safety and health in these times. And thank you all for participating. So, Richie, did you have any other final comments?
I actually don't, Robert. I just echo your comments in thanking everyone, including our audience. And if you have any other questions, don't hesitate to reach out to us, to reach out to our researchers and the panelists today. And we look forward to seeing you again in the future and sharing more of our results as well as our progress. Thank you.